Hi, my name's Abish. Welcome to my final year project presentation, Data Science for Diffuse Large B-cell Lymphoma, or DLBCL Stratification. DLBCL is a type of cancer, so I'll go into a little bit of cancer background first and set the scene for our problem. So in the body we have pathways, which are networks of these molecular interactions which help regulate our infection defense mechanisms. So changes in these pathways will cause a change in the functionality of these specific mechanisms. Cancer manipulates these pathways in order to control the mechanisms for its own survival, and for that reason, pathways involved in more than one mechanism become ideal targets for the cancer, because if it can successfully hijack these pathways, it gets access to more mechanisms, more resources in the body that it can utilize for its own survival. For this reason as well, the NF-kappa B pathway, because it's involved in two vital infection defense mechanisms, cell death and inflammatory response, becomes a common target across a lot of different cancers. So the cancer, once it develops in the body and manipulates the NF-kappa B pathway, hijacks the cell death mechanism, which causes cancerous cells to survive, and then also hijacks the inflammatory response mechanism, which suppresses the body's response to these cancerous cells. So it's a little bit of a catch-22 situation where you're getting these cancerous cells surviving and then the body being unable to fight back against them. So a key medical aim of all of this is to remove the cancer without affecting the normal healthy functionality of NF-kappa B. The issue is that it's very complex to develop a drug that targets the NF-kappa B pathway directly, since it's involved in two of these mechanisms, not just one, unlike a lot of other pathways. And so what researchers and clinicians did is they inverted the problem to look at the effects of the pathway rather than the pathway itself, and in doing so identified this gene called GAD45B, which was activated as a result of both processes. Now essentially, if we're able to block this gene off, depending at the level at which it appears for each patient, then we can block the cancer off successfully while still maintaining the healthy functionality of the NF-kappa B pathway. And that gets us onto our specific project goal, which is to stratify using GAD45B. So by stratification, I mean creating patient subgroups with different characteristics. By GAD45B stratification, I mean creating patient subgroups with different GAD45B gene characteristics. So investigating DLBCL, well, this GAD45B gene effect was initially identified in a different cancer called multiple myeloma. We're investigating it here in DLBCL essentially because if the GAD45B stratification can be successfully identified and verified in different cancers, the potential GAD45B gene drug becomes multi-purpose and so is easier to get to market. So we look specifically at 775 DLBCL patients. We produce subgroups using gene expression data. So gene expression data essentially measures the activity levels of the genes in each patient. So what we essentially do here is group together patients to have similar gene expression patterns, and then we evaluate these patient subgroups using clinical data. Now, a key aim of all of this is that we want to ensure biologically robust subgroups. And what do I mean by that? I mean, we want to have patient subgroups that are reliable and reproducible if they were to be done using different cohorts or in different studies. And an issue is that if we enforce these GAD45B differences, it could prevent some of this biological robustness. So we opted for the solution in this project of not enforcing these differences, instead making the project an unsupervised analysis task. So the first thing to do once we had our data was to check whether an existing stratification in DLBCL, not GAD45B based, already satisfied our GAD45B requirements. So the most well-studied stratification in DLBCL is the cell of origin, COO stratification, splits patients up into the ABC, GCB subgroups. If they don't fall into either of those, then unclassified. But this didn't give us any conclusive results for GAD45B stratification and was further motivation for us to go on and produce our own subgroups. So in developing our method, we had two main requirements. The first is that with our gene expression data, we have an enormous amount of genes, over 13,000. And so what happens quite often is that you get a lot of irrelevant genes that mask the smaller subset of important ones that you actually care about, that allow you to differentiate between patient subgroups. And so we need to be able to deal with these irrelevant genes in our method. And secondly, as mentioned before, we need to ensure biologically robust subgroups. So we looked at 3 key papers in the field and looked at how they solved similar problems and developed our final method from them. The first step of which is gene filtering to get rid of some less important genes, taking us from 13,000 to 1,500 genes, our core set. And from this core set, we apply our second step, which is a compound clustering method called NMF consensus clustering, to create our N patient subgroups. Now, the NMF part of this algorithm is quite crucial, so I've just got a little diagram on the right-hand side to explain it. So given the initial data matrix, the NMF algorithm will summarize that into latent variables called metagenes, and we find the patients most associated with each metagene, and then assign these to a patient subgroup. So essentially, if we have N metagenes, then we're also going to have N patient subgroups. And the NMF ensures that these key patterns are identified in our data and allows us to produce our patient subgroups. The consensus clustering then ensures that our patient subgroups are robust, because essentially it averages out the result of the NMF algorithm with different random initializations. So doing this, we found that our most statistically robust results came with a stratification of 4 main subgroups and then 1 smaller unclassified subgroup. And we were successfully able to find the GAD45B stratification with our criteria on these subgroups, which gave us reasonable evidence for further clinical study. So to summarize what we did in this project, we devised a stratification method and subsequent stratification for DLBCL patients based on a potential drug target, the GAD45B gene. And then going forward, what we want to do is gain a more systems level approach. So in the short term, that means potentially integrating other pathways other than just the NF-kappa B pathway. In the long term, it means getting more patients and performing a multi-omics analysis. And so by that, I mean, what we did in this project, we considered gene expression data, which, depending on how you view it, either falls into genomics or transcriptomics. But if we can get the full range of multi-omics data and integrate them effectively, then we can gain complete biological networks to describe each patient's subgroups, and so can give them even more targeted and, more importantly, more effective treatment. So just to summarize this, just to finish off, this project drew on the knowledge of a few different fields. So I want to say thank you to the following people for helping me: Elsa, Richard, Natalie, Guido and Dario, thank you, and thank you for listening.